This is a production of Cornell University.
Okay, thank you, Bill, for that very nice introduction. Thank you all for coming in person. And yeah, so today I'm going to talk mainly about long encoding RNAs or various subcategories of long encoding. Okay, so most of us are familiar with the central dogma. We know that we get some kind of a signal or a cue from the environment, which elicits some kind of change in the DNA, which produces an RNA. Transcription produces an RNA, and then translation translates that RNA into a protein, which for most of us, for many years, this protein was what we assumed was contributing to the response. But in the past, let's say 20, 30 years, we know a lot more about the non-coding RNA portions of the transcriptome, including long encoding RNAs and small non-coding RNAs. And indeed, there are other things that can help regulate responses, including post-transcriptional RNA modifications that are added to these different classes of RNAs that all contribute to regulating the response to changes in the environment for this poor little Arabidopsis plant. Now, the lab is broadly interested in all things RNA, but in general, or more specifically, we're interested in long encoding RNAs, post-transcriptional RNA modifications, I alluded to here with the yellow lollipops, changes in RNA abundance or stability, and how that just changes in transcriptional regimes help plants respond to stress or respond to changes. And then, of course, RNA compartmentalization within the cell. So here I'm talking about stress gradient. And we work in a variety of systems. The good thing about most bioinformatics is that you can do it in a number of systems. You just have to be working with experts who know something about the plant that you're working with. But in general, our home base, so to speak, is the Brassicaceae. Okay, so today, long encoding RNAs. Now, what are long encoding RNAs? Obviously, they're non-coding, and they're long, so greater than 200 nucleotides. In general, they don't have an open reading frame greater than 100 amino acids. But we heard from last week from Polly that there are a whole bunch of transcripts in the cell that encode or have short open reading frames. In fact, that will be a subject of a few of my slides later on. But in general, lincRNAs are long, no open reading frame. They can come from anywhere in the genome. We kind of categorize them based on where they are coming from in the genome. Most are Pol2 transcribed and typically display poor sequence confirmation. In terms of function, lincRNAs primarily function inside the nucleus. Perhaps the most famous long encoding RNA generally isn't even thought of as a lincRNA because it's so famous. It's the telomerase RNA. And the telomerase RNA functions in the nucleus to maintain chromosome ends. It's functionally conserved across all eukaryotes. It's missing in the monoorganism Drosophila, but it's present in almost every single lineage, every single species. So it's a highly functionally conserved long encoding RNA. There are also other lincRNAs that function to modulate chromatin dynamics. This can be compaction or opening up of chromatin. Famous example in plants is the Apollo lincRNA that forms these chromatin loops that help to suppress or to regulate pinoid transcription and actually modulates how root branching occurs. Perhaps maybe an even more famous lincRNA is actually an antisense lincRNA called COOLAIR that regulates the FLC locus and modulates flowering in Brassicaceae. And these COOLAIR represents a... Okay, back to lincRNAs in the nucleus. So COOLAIR, as I mentioned, represents this class of lincRNAs that modulates epigenetic states. So we're talking about histone or DNA methylation. And then there's another highly conserved lincRNA called PID1, which actually binds to transcription factor and targets transcription factors to different locations. I'm going to come back to PID1 and telomerase RNA in a moment. And then, of course, we have other lincRNAs that perform other functions inside the nucleus. All of this is to regulate either transcriptional dynamics or to modulate a half-life or maturity of messenger RNAs, with, again, the sole exception of which ones. Out in the cytoplasm, there are fewer characterized functions for cytoplasmic lincRNAs, But perhaps the first lincRNA identified in plants, E940, identified in soybean, or medicago in soybean, is actually binds to a protein in the cytoplasm and can be cell-to-cell mobile. So, example of mobile lincRNAs. And then, of course, there's another lincRNA in the cytoplasm, another example of lincRNA in the cytoplasm that actually sequesters microRNAs and decoys and pulls them away from their intended target. So this long list of functional archetypes represents the best of what we know about plant lincRNAs, but also represents most of what we know about plant lincRNAs. Aside from this list, there's maybe 5 or 6 other well-characterized lincRNAs. They fall within these functional archetypes. The list of characterized lincRNAs in Arabidopsis is about 20 to 25. So I would argue that the extent of what we know, the extent to which lincRNAs contributes to critical cellular processes is poorly understood, largely because we don't even know what's there. We haven't known what's actually transcribed, what's present in the genome. So here I'm showing you a phylogeny of some of our favorite plant species. And by favorites, I mean these are the ones that we study and we are most deeply sequenced. And then, of course, there's a few basal eukaryotes here at the bottom. And in terms of lincRNAs that are present, that are available and annotated in the genomes of each of these species, of course, Arabidopsis has the most, but it's nothing in comparison to humans and mice. And this is largely due to the Human Mouse on Code projects, which went through and attempted to annotate all functional elements of those 2 genomes. In contrast, where lincRNAs have been identified in plants, we have different criteria and experimental evidence used in that identification effort. And so now we start to be comparing apples to oranges when we're looking at rice or Arabidopsis lincRNAs. So we're not even making fair comparisons when we want to start looking, performing function. Now, lincRNAs can be identified from RNA sequencing data. And when we look at the amount of available RNA sequencing data as of 2018, it starts to become more equivalent between plants and humans. At some point, you have a wealth of RNA sequencing data. You don't really need any more. And you can start identifying long-read coding RNAs from all of this data. And so really that's what we decided to do. We've decided to go through and start identifying RNAs using the same methods, the same criteria. And we started this in the Brassicaceae. Now we went through and we processed probably way too much RNA sequencing data in retrospect. There was something to the tune of 20 terabytes or 20 terabases of RNA sequencing data, which if you know anything about sequencing data, that's a metric load of sequencing data. Most of it is from Arabidopsis, but we had a good amount from Brassicaceae. We processed all of this sequencing data. We remapped everything, identified transcripts using this information, and then figured out which of those transcripts looked like lincRNA. For all 4 of these species, we identified thousands, tens of thousands of lincRNAs, about 20,000 in Arabidopsis and more in each of the other 3 species of interest. And then we performed basic characterization and putative functional assignment. And so just some general take-homes from the general identification is that we used short-read data, we used long-read data, some of our own long-read data, and to identify lincRNAs. And then this process kind of helped us. We went into this a little bit naive and thinking that it would maybe be very straightforward and super easy, but we developed additional metrics that we believe will help us filter out spurious transcriptional artifacts in future identification. But let's talk about what we've done for Arabidopsis and the other Brassicaceae. We've really gone from a scenario like this where, okay, we've got a lot of protein coding genes, about 24,000 or so. We have some small RNAs, and we have some lincRNAs, and we have some functionally characterized lincRNAs. And then we've gone to a scenario more like this, where all of a sudden, everything's transcribed. Who cares? Like, what does this even mean? How do we make sense of this? And so now we have the what I call the false summit of lincRNA identification, where we've been doing a lot of work. We've been going up this mighty, mighty mountain, and we reach a peak, and then we realize that we have a whole lot left to do, because now we're going from identification. So now we really need to start making, we need to transition from identifying transcripts to figuring out what those transcripts are. So we can make some of those inferences based on the analyses and the work that Kyle and Leon and Kaylin and others did in the lab. And one of the things that we can use to help us is to simply determine like which of these loci, which of these lincRNAs are conserved. I mentioned that the telomerase RNA is highly conserved. It's both sequence conserved within families, and it's structurally conserved. You can use multiple sequence alignments to infer lincRNAs, telomerase RNAs and other families across the land plant lineage. So they're actually pretty easy to identify once you know what you're looking for. And in addition, the HID1 locus, the HID1 lincRNA is also very sequence conserved across land plants. So the question is, are there other telomerase RNAs? Are there other HID1s? Are there other highly conserved lincRNAs? And what I'm showing here is again, a phylogeny of Brassicaceae, along with Terranea hasseleriana, so a representative of the sister family Climaceae, and simply asking at the sequence and symphony level, do we see conservation of any of our 20,000 Arabidopsis lincRNAs? Very, very straightforward, sequence and symphony. And we see that for about 250 of these 20,000 lincRNAs, we see that there is sequence conservation that we can detect, and these are coming from synthetic loci. These lincRNAs are probably doing something important. They're conserved across a very long distance, much more so than the bulk of the Arabidopsis lincRNAs. About 43% of the Arabidopsis lincRNAs are species specific. So this is a big contrast to what we're seeing here. And if we go and we generate multiple sequence alignments and we look at structural data from in vivo structural data, then we can actually infer and we can determine that some of these lincRNAs are actually conserved outside of Brassicaceae, and in fact in much deeper lineages, such as HID1 or the telomerase RNA. So these are obviously, if you're cherry picking your data and you want to, or cherry picking your candidates for functional analysis, these are a strong place to start. And as an example, this is a collaboration with the Boyce and Gregory labs. We identified a lincRNA that was conserved across Brassicaceae. It was actually originally named Cobra. It's covering up the cute name. So link Cobra, so conserved in Brassica. So hence the nice little schematic that Marianne, the lead author, came up with. But this long-range coding RNA, link Cobra, it's about 450 nucleotides. And you'll see from this alignment that there are two regions of strong sequence similarity. And these two regions correspond to two snow RNA-like motifs. So they're not full snow RNAs, but they share sequence similarity. And in addition, these are protein binding sites that the Gregory lab identified through PEP-seq. So we have some information straight off. One, conserved lincRNA, and it's protein binding, long-range coding RNA, or it's bound by protein, which isn't surprising because all RNAs are bound by. Okay, so the Boyce lab and the Gregory lab, Marianne and Kyle both created different tDNA or CRISPR immunogenesis lines. Here we're showing both of these, and you see that they're not only delayed growth, but there's also decreased in seed germination after 48 hours. So these mutants for this long-range coding RNA are having an impact on plant development. And going back to the protein binding aspect, performing CHIRP-MS, we see that this RNA, so CHIRP-MS is essentially you're pulling out the RNA from in vivo, and then you're performing mass spec on any proteins that are bound to that RNA. You see that there's a whole set, there's something like 70 or so proteins that are bound, and most of these are either they're involved transcription factors or other RNA binding proteins. Now, the reason that we initially, or the reason that we went with this lincRNA looking at its protein capacity rather than whether or not it was a cis or trans regulator was because this, from all the transcriptomic data that we had, we could tell that the expression of this lincRNA wasn't correlated with the expression of the adjacent protein coding genes. So we thought that its function was most likely through whatever proteins it was interacting with, and not through some kind of cis or trans regulation. Now, getting back at those cis or trans regulators, we know for at least a subset of them that those link RNAs should likely interact with the RDDM pathway or other modifiers of epigenetic states, or chromatin modifiers. And so there's a whole lot of data about available for small RNAs mapping back to, so there's a whole bunch of small RNA data, as well as small RNA data produced from mutants within the RDDM pathway. And so we can test whether any of our link RNAs look like either, well, if they look like Apollo or if they look like Coué, any type of link RNA that would regulate chromatin. And so first, we simply took small RNAs from a database that the Myers lab curates, and we asked if those small RNAs overlapped with any of our link RNAs. We see that about, in the subset plot, we see that about 4,000 of our Arabidopsis link RNAs overlap with small RNAs. And of those, about 2,000 in the tissues for which we had data, 2,000 of those link RNAs had a dynamic relationship with small RNAs, implying that they're either being regulated by or are helping in the production of small RNAs. Now, the RDDM pathway is traditionally thought of as regulating transposable elements. And so the next question would be whether any of these link RNAs are actually just transposable elements in disguise. And so we went through and we re-annotated all the transposable elements, or we re-annotated the Arabidopsis genome for transposable elements. And then we asked if any of those transposable elements overlapped our link RNAs. So to consider overlap, it would be 50% or more of the TE overlapped with the link RNA. So about 1,000 of the original 20,000 Arabidopsis link RNAs overlap to some extent with the transposable element. But of those, so now these are called TE-associated link RNAs. Of those, only 170 of these 4,000 are TE-associated link RNAs, suggesting that these link RNAs, a large number of these are acting more like Apollo. They're active modifiers of chromatin dynamics rather than being actively suppressed by the RDDM pathway. We can also get or assess whether or not our link RNAs are being activated in response to changes in the environment. So are they stress sensors or are they developmental sensors? And here I'm just showing you stress data. This is essentially asking if any of our link RNAs are differentially expressed in response to any of these 5 stresses. And you see that there's a decent number. There's actually not as many as we initially thought, but there's about 1,000 or so link RNAs that are differentially expressed in response to one of these 5 stresses. And we know that a subset of these are ADA dependent, so we can further narrow down the regulatory pathway for these link RNAs. And so if a link RNA is stress responsive, that doesn't necessarily mean anything in and of itself. There are a lot of genes that get activated or repressed during heat stress, for instance. You know, it's every paper you read, it's going to be 3 or 4 or 5,000 genes are differentially expressed. So to ask whether or not these link RNAs are actively regulating the acting as cis or regulators, we look to see if their expression is changing, the correlation in their expression with adjacent genes is changing in response to the environment. So here I'm just showing you a very simple cartoon of a long non-coding RNA and its surrounding genes, gene X and gene Y. And we'd imagine a scenario where when the link RNA is upregulated, actively suppressing the adjacent gene, gene X, and so you see a decrease in expression. Now, we would expect that correlation, so this would show up as a negative correlation in expression anytime we found a tissue where the link RNA was upregulated. But we would also expect to see scenarios, if this was a specific correlation, we would expect to see scenarios where the correlation changes, the dynamics change. So we went through and we asked how many of these link RNAs, these stress-responsive link RNAs, were correlated with their adjacent protein coding genes or adjacent genes. And then of those, how many of those either positive or negative regulators, how many of those correlations changed? And then it's the change that we're interested in, because we think that that's probably a specific form of regulation. So again, a large number, I think it's, again, about 1,500 or 2,000 link RNAs are probably acting as cis-regulators. And we can further look into those using either transcription or epigenetics to get a better understanding of that relationship. We can also look at the correlation or the similarities in expression and develop these large co-expression networks, like I'm showing here, where this network is actually showing representing a whole bunch of transcripts that are similarly expressed. So they fall into the same module of transcript. And in this case, the unifying feature, unifying pattern for all of these transcripts is really this peak early on in wounding. And so these genes would be what we would consider wounding responsive. And in fact, they would probably, I would call them responsive, like they would be early wounding. And if you look at the kind of clusters of genes that are interacting, particularly transcription factors that are involved, it all makes sense with something that would be responding to wounds. You have jasmonic acid responsive genes, and you also have wounding responsive genes. Again, all thanks to the massive amounts of annotation that's already present for Arabidopsis. And so we can use all of this information to say, okay, we have 5 link RNAs in this module. What are they connected with? Whom are they interacting with? And can we infer, can we make a functional hypothesis based on those interactions? And because we're in Arabidopsis and we have a whole bunch of DAP-seq data and other ChIP-seq data for the transcription factors, we can make assumptions or hypotheses about which specific transcription factor is actually regulating these RNAs during the wounding response. So normally this would just be a co-expression network, but when you can overlay aspects such as transcription factor binding, then all of a sudden you can start to call this a regulatory. So again, about 2,000 link RNAs interact with mRNAs in a stress or development associated co-expression network. So I've just told you 3 or 4 different scenarios, different types of data that we can use to make functional hypotheses about our link RNAs, but that's still, it doesn't get you to the function, it's just a hypothesis. So we have all this evidence to support function. We have interesting features or associated modules that we would like to test. So now we need to start doing some reverse genetics. So again, Arabidopsis is really useful. There's a lot of insertion lines available. CRISPR is very easy in Arabidopsis. So we can do this and we can combine it with high throughput phenotyping or very simple, very rapid screens, such as germination screens, to then succeed at functionally annotating our long-read coding RNA. So let's go through a couple of examples where we're beginning to do just that. So here we're looking at, we're using the germination screen. This is work done by Kalin. And we're simply asking link RNAs that are expressed and fit into this module of germination-associated transcripts. Are they, if we mutate them, are they associated with a germination delay or germination phenotype? And it's not, I am not a germination expert by any means, but this is just an easy and rapid phenotype to go after. We want to validate our approach, and we're doing this with link RNAs with a phenotype that's easy to score. And easy to score is simply this. We see very little germination or delayed germination for the link RNA mutants, where we see germination is just fine for the wild type. And so Kalin screened through 14 different link RNAs that fit into this module of germination-associated transcripts. And we see a decrease in germination rates for 11 of those 14. Now, as you see, the germination decrease varies quite a bit. Wild type, maybe wild type's not even shown. I think wild type's here. But as you can see, there's quite a range, but there are some that have very strong decrease in germination. So then Kalin, part of her dissertation work has been to go through and assess what is actually occurring with some of those long-encoding RNAs. So these LGD1 or LGDs, this one, sorry, LGD1. So we believe it's a cis-regulator of PAP2. And the reason that we think that that's occurring is partly shown in the schematic here, where we have LGD1 is this box here. There's a pseudogene just directly downstream of LGD1 going in the opposite direction. And then there's this protein coding gene called PAP2. So PAP is a phosphatidic acid phosphatase. It's important for germination. It's important for lipid biosynthesis. Now we had two independent tDNA insertion lines that disrupted LGD1. Oh, and I should also mention that both LGD1 and PAP2 show expression, high expression in germinating seeds and early seedlings. However, the pseudogene does not. And so just to remind you again of the phenotype, both of these alleles, we see decrease in germination, so slower germination. And then once the seeds do germinate, they're typically, or sorry, there's reduced germination. And then for the seedlings that do germinate, they germinate at a much slower rate. And then some of those seedlings actually essentially abort and die. Now, some other things I've marked here on the schematic is that LGD1 overlaps two enhancer regions. And one of these enhancer regions contains a MIB107 transcription factor binding site. Now, MIB107 is interesting because MIB107 regulates other genes associated with germination. In fact, MIB107 is critical for proper seed coat formation, or it regulates genes that are important for proper seed coat formation. So the fact that MIB107 is here and not disrupted by the two Salk tDNA insertions that we have here, but it is found within the transcribed region of LGD1 is highly reminiscent of the Apollo story, where Apollo actually forms this chromatin loop and helps to regulate the expression of the downstream gene. But when you disrupt Apollo, you disrupt expression of the downstream gene Henry. So this is the hypothesis we're working with, but obviously Kaylin has quite a bit more work to do to flesh this out. Now, the last story that vignette that I want to tell you about is about some of these lincRNAs that actually contain short open reading frames. Now, a lincRNA with a short open reading frame is probably not a lincRNA, right? So we don't necessarily know that these short open reading frames are producing peptides. We have evidence to suggest that, but we don't know that the peptide is the functional unit, is what I'm trying to say. So ENOD40, this lincRNA that was characterized by Martin Crespi in the late 80s, also has two short open reading frames within it. But the functional unit of ENOD40 is the RNA itself, not those two proteins. So we have a whole bunch of lincRNAs. I think there is something like 150 lincRNAs that we identified that have these open reading frames within them. And here we're just characterizing what the lincRNAs look like, or sorry, what the open reading frames look like, what kinds of proteins will they produce. And here, this was just for the paper, we were just describing that there were more found within the airport, the older annotation, or fewer of these found within our annotation. That's not as important. What's really critical here is that you can see there's a lot of really short potential peptides that are being produced, for which we have evidence from riboseq data. So this is actually in collaboration with Boyce. And for some of these, we also have evidence from mass spec data from OLA. So each of these open reading frames, or sorry, each of these peptides produced from a short open reading frame, we then asked, are these just fragments? Are these fragments of known proteins? Or are they something novel peptides? And probably hard to see, but the green circles, which there aren't very many of them, those are the peptides that actually corresponds to a known, there's only 10 for you in the back, very few, of these peptides that are actually look like a known protein. Some of them have already been annotated or look like hypothetical proteins. So that's another 47. But a hypothetical protein, again, in Arabidopsis doesn't mean anything. It just means maybe we have mass spec data for it, maybe we don't. But then most of these peptides are unique. There's no similarity to them in any other protein database in any other eukaryote. Again, doesn't necessarily mean anything, but it is interesting. What might these peptides be doing? And so we went through and we decided, let's take the lincRNAs that contain short open reading frames that are conserved, highly conserved, and let's see if we can test, come up with some way, a genetic and a molecular way of testing for function. So first, again, the standard germination assay for one of these peptides, one of these supposed to be peptide with germination defect, PGD. One of these, it's expressed early on in seeds or in seedlings and in seedling roots. And so this was the one that Alyssa tested on plates to see if it interfered with germination. And it did. So over a period of 4 days, most or all of the Columbia, the wild type seeds germinated, but there was significant delay for the insertion line for this particular peptide, this particular SORF containing inRNA. So okay, that's for the lincRNA, the peptide that's found where we would expect a germination defect. But what about these others where they're not necessarily expressed in seedlings or in germinated seeds? So like, what's the phenotype that we would look for with them? And so with this, we collaborated with Magda to use the phenorigs to figure out if there's any kind of growth delay for these particular mutants. And so we grew each of these mutants under the phenorig. We took images every 30 minutes, took that over a period of 3 weeks. You see the plants slowly growing. And then with using plant CV, we go through and figure out what the growth rates are for these mutants compared to wild type. Of course, all in like randomized screening, randomized plot design. And what we see, so this was work by Alyssa and an RU student over the summer, is that these mutants display differences in growth rates. So we have a couple of 2 mutants that show decreases in growth rates. And then interestingly, we have 1 mutant that actually shows an increase in growth rate. Okay, so now what I'm not showing you is all the peptide containing lincRNAs that don't have phenotypes. So now we have some interesting candidates. We have a reason, we're using this high throughput screen to figure out which of these peptides or these SORF containing lincRNAs might be of interest. And then we need some kind of additional high throughput way of figuring out like where we go, what we target the function, what developmental stage, what stress, et cetera. So in collaboration with Ola, we're using a protocol called LIPMS, Limited Proteolysis Mass Spec. And how this works is you take protein extracts from your tissue of interest, wherever you think that your peptide is expressed. You extract, or you take that protein extract and you mix it with a synthesized peptide. So we went to GenScript and we asked GenScript to synthesize the peptides for, I think, 10 or 11 of the peptides that we're interested in. We mix it with the extract. And what happens, what potentially we hope is happening, is that our peptide will interact with a protein or a set of proteins and will change the conformation of those proteins. Then we perform limited proteolysis. We get different size of peptides as a result from that proteolysis. And what we hope is that the addition of the peptide is going to change the pattern of proteolysis, meaning that that peptide has actually interacted with one or more proteins in this extract. And so what this would look like is it's almost like a volcano plot from transcriptomics. You would expect to see some kind of increase, significant increase in the number of peptides that are changing or moving, I say moving, that are different between your control and your peptide setting. So let's just show you the peptide that Alyssa's looked at so far. So AT1G 06113 produces a very short peptide. So it's a 27 nucleotide long open reading frame. So it's a 9 amino acid peptide, highly conserved. In fact, it's conserved across all Brassicaceae. And the nucleotide, sorry, the amino acid sequence is completely conserved across all Brassicaceae. Oh, and other important things is that we know that this transcript, the transcript itself, is responsive to oxidative stress, as well also is differentially expressed during hypoxia. So we have some expectations about what this protein might be interacting with. So when Alyssa added this peptide to extracts, she saw that there were 23 peptides from 18 proteins that were differentially abundant between mock and peptide treated samples. We then can go through and just sanity check and make sure that the targets are actually representing proteins coming from transcripts that are expressed in the same tissues. If they're not present in the same tissue, then they can't be interacting. And we see that, so here on top is our peptide, sorry, the transcript encoding for our peptide. And then you see that most of these proteins are, the transcripts encoding for all the interacting proteins are also expressed in the same tissue. And if you look at if you perform a simple Go enrichment of the proteins present here, we see that there's enrichment of oxidative reductase activities, small molecule binding, but more importantly, responsive stress, abiotic stimulus, and chemical stress. So things that almost look like what we're seeing with the transcript itself. Okay, so with that, some final take-homes. We've identified a lot of lincRNAs. We identified lincRNAs in 4 absolutely critical species within the mustard lineage. And we are in the process of functionally characterizing, really screening through and figuring out which of the lincRNAs we think are low-hanging fruits to perform functional analysis. And then we're also moving our annotation efforts to other species. So there's a lot of lincRNAs that have been annotated, or a lot of species for which lincRNAs have been poorly annotated, and we want to fix that. And yes, so with that, thanks to a lot of people in the lab. And Xiaodan, the newest member in the lab, is going to be continuing on with the lincRNA story, working on a project that hopefully will encompass both plants and mammals, a large collaborative project. And with that, I need to thank Mark Beilstein, who's in the room, as well as other collaborators, Brian Gregory, Eric Lyons, Polly, Rebecca Murphy, and some RU students. And then, of course, the other heads of the Cerberus here at SCI, Ola and Magda, and then all the people here who actually contributed to the lincRNA itself. And with that, I'll take any questions. Questions? Yes, sir.
You showed us that the expression of quite a large number of them were affected by these stresses, cold and heat.
Mm-hmm.
I think you mentioned that might relate to ABA.
Yes.
And yet the drought number was really quite small.
Yeah, so it's all about sampling. So really it's, you're right that there are like heat and cold and drought, there are osmotic stresses, and we should see quite a bit of overlap. We do see, let me go back to that. Yeah, so this is what you're referring to. And the fact that there's very little overlap between this set here, which you would imagine would reflect all of the osmotic stresses. I think in part that represents the specificity of lincRNAs. I think that's the general response that I would give for that. Yeah, I can leave it at that. Yes. Mm-hmm.
And I'm trying to say like the genomic process of RNA responsive stresses. I think like epigenetic modeling of the genome is something that we're learning a lot of stuff. We started looking at the intersection of epigenetic marks and when RNAs get turned on and sterilized.
Yeah, so we haven't looked at specific cases. We have kind of a global, or we performed a global survey of the transcriptome and separating, protein coding genes from incarnates and transport development. But you're right, it would be really, it would be advantageous for us to go through and figure out for the ones that are particularly like stress responsive is what's initiating that transcriptional activation. And I think for us, it's just about finding the right paired experiments. So the other thing here to go back to your question is that the heat, cold, and salt are coming from the same experiments or the same, there's a little bit of batch effect involved in what's going on here. So the drought and the light are performed by different people and there may be issues with that. But to go to your question, you're absolutely right. We need to find the right paired experiments where they perform the epigenetic analysis at the same time as the stress. And that's there. It's just bandwidth. Yeah. Anyway.
So what do we know about IncRNA
outside of flowering plants? Does Fisco count? I just say, like, from a model system perspective. So HID1, the IncRNA that I said was conserved across land plants. So HID1 is present in Fisco. We know that you can take actually, no, they didn't do that experiment. We know very little. So I think so there's a couple of examples of deeply conserved IncRNAs. The telomerase RNA has been identified in all chlorophenic organisms and been functionally validated in a few. But other than that, I think one group has just done a characterization of IncRNAs in Fisco. But again, that's it for outside of transfers. Ciliates is where the telomerase RNA was first identified. So doesn't quite count.
That's it. Thanks very much.
This has been a production of Cornell University, on the web at cornell.edu.